Hello. Our research was about finding a polygenic mutation signature of metastatic breast cancer. So we have two types of tumor samples here. The red ones did develop metastasis, the green ones did not. For this class we did two splits. The bigger one we used to find some mutations specific for the positives. In the smaller split, we first removed the most difficult to allocate to any of the classes, and we end up from 62 to 50 from each class. For that we use random forest. So here is how we go back from 62 to 50. Random forest outputs a probability of belonging to the positive class, and probabilities close to 0.5 are weak probabilities. So these samples are most difficult to allocate to any of the classes. So we removed the 20% samples with probability closest to 0.5. So that's how we end with 50. With those 50 from each class, we found some of the mutations selected in the bigger set. For a particular split, we find this number of mutations, 49 eventually at the end, and we store those SNPs. But we are going to do many splits. This is just the results for one of them, and the results for another split. The number of mutations have changed, and we repeat this 10,000 times. So eventually we end with 10,000 selection of SNPs. For the first split it was 49, for the last one it was 63. And from these selections of SNPs we selected the 900 most frequent SNPs, most frequently reported. We call this our polygenic mutation signature. We use this signature to try to predict from new samples that the method has not seen whether they belong to the positive or negative sample class. But previously we applied random forest, same as before, to discard the most difficult to allocate samples, because we don't believe we have power to choose these ones. So with the remaining 80 from each class, there we found 83% accuracy in assigning samples to their class using this signature. Thank you very much for your time.